For decades, scientists have dreamed of precisely editing genes to cure diseases. This vision has driven countless hours of research and experimentation, pushing the boundaries of what we thought was possible in the realm of genetic medicine. This dream is now a reality with CRISPR technology. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a revolutionary tool that has transformed the field of genetics. CRISPR allows scientists to make targeted changes to DNA with unprecedented precision. By harnessing the natural mechanisms of bacterial immune systems, researchers can now edit genes with a level of accuracy that was once unimaginable. This groundbreaking tool holds the potential to revolutionize medicine. It is not just a scientific breakthrough, it is a beacon of hope for millions of people suffering from genetic disorders. It offers new hope for treating a wide range of genetic disorders, from cystic fibrosis to sickle cell anemia. Patients who once had limited options now have a chance at a better quality of life. CRISPR acts like molecular scissors, cutting DNA at precise locations. This ability to target specific genes is what makes CRISPR so powerful. It can cut DNA at specific locations, allowing for the removal of faulty genes or the insertion of new, healthy ones. This precision is key to its effectiveness. This allows scientists to remove, replace, or even correct faulty genes. The implications of this are profound, offering the potential to not just treat but actually cure genetic diseases. The implications of this technology are vast. Beyond treating genetic disorders, CRISPR opens up new avenues for research and development in various fields of medicine. It opens up new possibilities for treating diseases that were once considered incurable. Conditions like muscular dystrophy, Huntington's disease, and even certain types of cancer could potentially be treated with CRISPR-based therapies. CRISPR's potential extends beyond treating genetic disorders. It is also being explored for its applications in other areas of medicine, such as infectious diseases and personalized medicine. It can be used in various areas of medicine, including the development of new vaccines and the creation of more effective treatments for a wide range of conditions. This includes developing new diagnostic tools that can detect diseases earlier and more accurately, leading to better patient outcomes and creating more effective gene therapies. These therapies have the potential to not only treat but also prevent diseases, fundamentally changing the landscape of healthcare. The future of medicine is being rewritten with the power of CRISPR. As we continue to explore its capabilities, we move closer to a world where genetic diseases are a thing of the past and personalized medicine becomes the norm. Sickle cell disease is a debilitating genetic disorder that affects the red blood cells in the body. It is a condition that can cause immense suffering and significantly impact the quality of life of those who have it. It affects millions of people worldwide, with a particularly high prevalence in certain regions, including sub-Saharan Africa, India, the Middle East, and the Mediterranean. The global impact of this disease is profound, affecting not just individuals but entire communities. This inherited blood disorder is caused by a single mutation in the gene that codes for hemoglobin, the protein responsible for carrying oxygen in the blood. This mutation leads to the production of abnormal hemoglobin, known as hemoglobin S. Hemoglobin is the protein in red blood cells that carries oxygen throughout the body. In healthy individuals, hemoglobin allows red blood cells to remain flexible and round, enabling them to move easily through blood vessels. In sickle cell disease, the faulty hemoglobin causes red blood cells to become rigid and sickle-shaped. These sickle-shaped cells are less efficient at transporting oxygen and can cause blockages in blood vessels. These misshapen cells can block blood vessels, leading to reduced blood flow and oxygen delivery to various parts of the body. This can result in episodes of severe pain, known as pain crises, and can cause damage to organs over time. This leads to a range of serious health problems, including severe pain, organ damage, and increased risk of stroke. Pain crises can be excruciating and may require hospitalization for management and relief. Sickle cell disease predominantly affects people of African descent, but it also occurs in other ethnic groups. The high prevalence in African populations is due to the genetic advantage it provides against malaria, a common disease in those regions. This highlights the disparities in healthcare access and research funding for diseases that disproportionately impact marginalized communities. Many individuals with sickle cell disease face challenges in accessing adequate medical care and support. The development of effective treatments for sickle cell disease is crucial for addressing this health disparity. Advances in medical research, including gene therapy and new medications, offer hope for better management and potential cures for this debilitating condition. Recognizing the urgent need for a cure for sickle cell disease, Vertex Pharmaceuticals and CRISPR Therapeutics joined forces. Their goal was to develop a CRISPR-based therapy that could potentially cure this debilitating genetic disorder. Vertex Pharmaceuticals brought its expertise in drug development and commercialization to the partnership. CRISPR Therapeutics contributed its cutting-edge CRISPR gene editing technology. This collaboration marked a significant step towards translating CRISPR's potential into a life-changing therapy. The two companies focused their efforts on developing a therapy that could correct the underlying genetic defect responsible for sickle cell disease. They aim to develop a one-time treatment that could potentially free patients from the burden of this lifelong disease. Section 4. Casgevy, a triumph of science and hope. After years of rigorous research and development, Vertex Pharmaceuticals and CRISPR Therapeutics achieved a historic milestone. In December 2023, the US Food and Drug Administration, or FDA, granted approval for Casgevy. This marked the first ever approval of a CRISPR-based gene editing therapy for any human disease. Casgevy's approval represents a paradigm shift in medicine. It demonstrates the potential of CRISPR technology to cure genetic diseases. This groundbreaking therapy offers renewed hope for patients with sickle cell disease and their families. The FDA's decision was based on compelling clinical trial data. This data showed that Casgevy was remarkably effective in reducing or eliminating the severe complications associated with sickle cell disease. Section 5. The journey of treatment from lab to life. The Casgevy treatment process involves several key steps that showcase the intricate interplay between laboratory science and clinical care. This journey is a testament to the advancements in medical technology and the dedication of countless researchers and healthcare professionals. The journey begins with the careful extraction of the patient's own bone marrow stem cells. This procedure is meticulously performed to ensure the highest quality of cells are harvested, as these cells are the foundation of the entire treatment process. These stem cells are responsible for producing all the blood cells in the body, including red blood cells, white blood cells, and platelets. Each type of blood cell plays a crucial role in maintaining the body's overall health and functionality. Once the stem cells are collected, they are sent to a specialized laboratory. This lab is equipped with state-of-the-art technology and staffed by experts in the field of genetic engineering and cellular biology. In the lab, the magic of CRISPR gene editing takes place. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a revolutionary technology that allows scientists to make precise changes to the DNA of living organisms. Using the CRISPR-Cas9 system, scientists precisely target and modify the BCL11A gene in the patient's stem cells. This gene plays a significant role in the regulation of hemoglobin production, and its modification is crucial for the treatment's success. After the gene editing is complete, the modified stem cells are carefully multiplied and prepared for reinfusion into the patient. This step involves growing the cells in a controlled environment to ensure they are healthy and viable for the next phase of treatment. This crucial step requires specialized medical expertise to ensure the safe and effective delivery of the edited cells back into the patient's bloodstream. The reinfusion process is closely monitored by a team of healthcare professionals to ensure the patient's safety and the treatment's efficacy. Following the reinfusion, the patient enters a critical recovery phase. During this time, the patient's body begins to accept and integrate the modified stem cells, which will start producing healthy blood cells. This phase is vital for the overall success of the treatment. As the patient recovers, they gradually regain their strength and return to their daily activities. The ultimate goal of the Casgevy treatment is to improve the patient's quality of life, allowing them to live free from the symptoms and complications of their condition. The journey from lab to life is a remarkable testament to the power of modern medicine and the relentless pursuit of scientific discovery. It is a collaborative effort that brings together researchers, clinicians, and patients, all working towards a common goal of better health and well-being. In the end, the success of treatments like Casgevy not only transforms individual lives but also inspires hope and progress in the broader field of medical science. It is a journey of innovation, compassion, and unwavering dedication to improving human health.